The objective of this practical is to determine the bleeding time in human body. Now we will understand the principle of this practical. Bleeding time is the time taken by a skin puncture to stop bleeding. It ranges between 1 to 6 minutes. Bleeding is influenced by vascular spasm. A vascular spasm is a sudden and brief tightening or constricting of a blood vessel. It reduces the amount of blood that can move through the vessel, sometimes even closing it completely and blocking blood from moving through. Next is platelet plug formation. It is an aggregation of platelets formed during the earliest stage of hemostasis in response to blood vessel wall and injury. Next is clot formation. Coagulation, which is also known as clotting, is the process by which blood changes from a liquid to a gel, forming a blood clot. Next is connective tissue formation. In living organisms, wound healing refers to replacement of destroyed or damaged tissue by newly produced tissue. Now, what is vascular spasm? When blood vessels are damaged, the circularly arranged smooth muscle in their walls contracts immediately. This reaction is called vascular spasm. This reaction reduces blood loss for several minutes to several hours, during which time the other hemostatic mechanisms go into operation. This vascular spasm is probably caused by damage to the smooth muscle, by substances released from activated platelets, and by reflexes initiated by pain receptors. Now we will understand platelet plug formation. Platelets contain vesicles. Within many vesicles are adenosine diphosphate, adenosine triphosphate, clotting factors, calcium ions and serotonin. Platelets also contain enzymes that produce thromboxane A2 and fibrin stabilizing factor, which helps to strengthen blood clot. Also within platelets is a platelet derived growth factor. It is a hormone that can cause proliferation of vascular endothelial cells, vascular smooth muscle fibers and fibroblasts to help repair damaged blood vessel walls. Immediately after a blood vessel is cut, contraction of blood vessel occurs from both myogenic and nervous reflexes. When platelets come in contact with damaged vascular wall, they become irregular and stick to the parts of a damaged vascular wall. These platelets become activated and secrete ADP and thromboxane A2. The release of ADP and thromboxane A2 activate and recruit other platelets, forming a mass of platelets called a platelet plug. Platelet plug formation occurs after vasoconstriction of blood vessels but before the creation of the fibrin mesh clot. which is the more permanent solution to the injury. The result of platelet plug formation is the coagulation of blood. Now, what are the requirements for this practical? In order to perform this practical, you will need blood lancet, spirit or alcohol swabs, stopwatch and filter paper. Now, what is the procedure of this practical? First, prick the finger taking precautions. The first drop which oozes out is not discarded. Start the stopwatch that has been earlier set to 0. At every 15 seconds, touch the drop of blood on the filter paper close to the edge of the filter paper till the blood stops oozing out. Non-spotting of the filter paper indicates that the bleeding is stopped. Quickly stop the stopwatch. The time on the stopwatch will indicate the bleeding time. Now, what is the clinical significance of this practical? A prolonged bleeding time is due to an inadequate platelets activity and failure of vascular contraction. The depth of the puncture or incision can be a source of error. This experiment helps in evaluation of platelets function and gives insights into different abnormal conditions such as thrombocytopenia, which is decrease in number of platelets, thrombocytopenia, which are defects of platelets, increased capillary fragility in old age, blood vessel defect, a blood vessel defect that affects how well your blood vessels transport blood through your body, and genetic platelet function defect. It is a condition present at birth that affects how well your platelets function, such as hemophilia, which is an inability of blood to clot causing severe bleeding. Here is one question: what causes decreased bleeding time?